Welcome back, students. In this session, let us learn about the structure and function of nucleus in eukaryotes. The nucleus is an important eukaryotic cell organelle. It functions as the administrative center and coordinates and controls the cell functions such as protein synthesis, metabolism, and cell division. The nucleus is the seat of genetic material, the DNA. The nucleus is the largest of all the cell organelles and occupies about 10% of the cell volume. Cell division starts with the division of the nucleus, and the rest of the cell divides only after nuclear division. The nucleus is a spherical organelle and is the center of nuclear transcription. Usually, there is one centrally placed nucleus in every cell, but some organisms have more than one nucleus in their cells. On the other hand, cells like human RBCs do not have a nucleus at all. There are no membrane-bound subparticles or subcomponents inside the nucleus. However, there are many nuclear bodies made up of unique proteins present inside the nucleus. So let's have a look at the nucleus structure and its function. Here is the structure of eukaryotic nucleus. It's a large organelle with a double membrane cover and has some important particles inside. As you can see the nuclear envelope, and the space between the nuclear envelope is called the nuclear pore. You have the genetic material in the form of chromatin. There's another large nucleolus, which is the heart of nucleus, and the nucleoplasm, in which all the nuclear materials are embedded, is what is commonly seen in nucleus. First, let us speak about the nuclear envelope. It is also called as the nuclear membrane. It is a double-layered cover consisting of two membranes, an inner membrane and an outer membrane, which separates the genetic material present inside the nucleus from the cytoplasm surrounding it. The inner and outer membranes of the nuclear envelope are separated by a perinuclear space, which measures about 10 to 15 nanometers. This membrane regulates the movements of macromolecules between the nucleoplasm and the cytoplasm. The outer membrane of the nucleus is continuous with the rough endoplasmic reticulum membrane. This layer has ribosomes distributed on it. So this RER is not visible in the diagram, but if you see the image here, you can see this is the nuclear envelope, and the nuclear envelope can continue as endoplasmic reticulum. If the endoplasmic reticulum has ribosomes on it, then it is called as the rough endoplasmic reticulum. The pores on the nuclear membrane are called as nuclear pores that allow free diffusion of certain molecules. The nuclear pores are made up of proteins called as nucleoporins. They help in facilitating the transport of only water-soluble molecules to pass through them, while the larger molecules like nucleic acids and bigger proteins are prevented from passing through the pores. Located at the place where the two nuclear membrane fuse is an 8-fold structure. This ring-like structure is called the nuclear basket and is present in every nuclear pore. The cell nucleus of the mammals usually contains 3,000 to 4,000 pores. The molecules that can pass through the pores are usually transported with the help of transport factors known as caryophorins. These caryophorins are of two types: importins, which help to carry the molecule into the cell, and exportins, which help to move the molecules out of the nucleus. While most of the caryophorins attach directly to the molecules, some of them use adapter proteins to interact with the cargo molecules that needs to be transported. Next is the nucleoplasm. So it is alternatively called as the karyoplasm. It is a gelatin-like substance inside the nucleus. It is made up of water, ions, and other molecules. The main function of these substances is to help the nucleus maintain its shape. It also supports the nucleoli. Hyaloplasm is the fluid part of the nucleoplasm, which helps in transportation of materials needed for the proper cell functioning. It also dissolves unwanted enzymes and nucleotides. Next is the nuclear lamina. These structures provide mechanical support to the nucleus. These are a network of cytoskeletal structural components called intermediate filaments. So the nuclear lamina is composed of lamin proteins, and they also anchor the chromosomes. Next, moving on to chromosomes. We've already learned in detail about chromosomes. The nucleus is the seat of this genetic material, that is DNA. They are organized into structures called as chromosomes. So this number of chromosome is very specific to every species. Say, for example, in human beings, we have 23 pairs of chromosomes, 22 autosomes and 1 pair of allosomes. So DNA is the genetic material of the cell, and we know that it is a double helical structure. So the long DNA strands wrap around histone proteins and form a structure that resemble beads on a string. So this is what is referred to as nucleosome. These nucleosomes fold into a compact, dense particle called as chromatin fiber. The chromatin reinforces the DNA molecule, and it plays an important role in regulating gene expression and protecting DNA from damage. The chromatin fibers coil and condense to form what is known as chromosomes. Other than these major components, we also have subnuclear organelles. So the three important subnuclear organelles are nucleolus, Cagil bodies, and speckles. Nucleolus is a dense substance which is found inside the eukaryotic nucleus. They are not bound by any membrane. Nucleoli are responsible for the manufacturing of ribosomes, the protein producing structures which is found in eukaryotic nucleus. The number of nuclei is specific for each species. Nucleoli disappear during cell division and appear back when the chromosomes are arranged into the nucleolar organizing regions after cell division. Next, moving on to Cagil bodies and speckles. These are spherical, non-membranous bodies found inside the nucleus. The possible function of Cagil bodies is to modify RNA after its transcription. Speckles or interchromatin granule clusters are abundant in the pre-RNA splicing factors. Now, what is the function of nucleus? It is a place where genetic transcription happens, and the primary function is to mediate DNA replication during the cell cycle and to control the gene expression. Apart from this, the other functions include compartmentalization of the cell. So the nuclear envelope gives a definite structure to the eukaryotic nucleus, and it separates the contents from the rest of the cytoplasm that encloses the nucleus. The nuclear envelope prevents the translation of unspliced mRNA by separating the nuclear process from the cytoplasmic process. The next important function is the gene expression. So DNA transcription occurs inside the nucleus, and the DNA transcription involves the unwinding of DNA double helix, synthesis of RNA, the coiling of DNA, and the actual transcription process. The next important function is nuclear transport. So transporting various molecules in and out of the nucleus through the nuclear pores is an important function of the nucleus. Important proteins are carried into the nucleus with the help of importance, and molecules like RNA are transported out of the nucleus with the help of exportins. Next function is assembly and disassembly. During cell division, the nucleus is integrated and disintegrated to divide the contents and the genetic material. The cell nucleus is also associated with programmed cell death or apoptosis. The nuclear lamina and the nuclear envelope dissolves and then formed again during the cell cycle. Duplication of the DNA and RNA replication takes place in the nucleus. Thus, nucleus integrates and disintegrates various structures during the different phases of cell cycle. The next function is pre-mRNA processing. So this mRNA, before getting transported to the cytoplasm, is what is called as the primary transcript or the newly synthesized mRNA. This goes through post-transcriptional modification, PTM. This happens inside the nucleus, and it involves a series of biological processes. Next function is ribosome biogenesis. A part of ribosome biogenesis takes place in the nucleus. Over 200 different proteins work in coordination to synthesize the 4 eukaryotic rRNAs, which then together with the ribosomal proteins synthesize the pre-40S subunit and the pre-60S subunit. These subunits are then transported to the cytoplasm for maturation. So to quickly summarize, the largest organelle and the seat of genetic material, that is the nucleus, is a very important organelle with clearly defined structure and function. Eukaryotic nucleus has evolved as a very important organelle in multicellular forms of life. The nucleus is definitely a feature of an advanced form of life and supports the complex functions of the cells. Nucleated cells have many advantages over those without a nucleus. Thank you.